So hi, hello and welcome. Microbehunter here, Oliver here, and welcome to another episode of Microbehunter Insights. Nearly forgot the title of my own channel. What am I going to show you today? Today I'd like to put white blood cells, some of my blood, under the microscope, and I would like to show you some staining reactions to stain and to make the white blood cells visible, to stain the white blood cells. And to give you a short overview of how I'll be doing that, short overview over the materials. I want to use two types of stains for making the white blood cells visible. One of them I have in this bottle here. I should have labeled it. It's methylene blue. It's known as also as Löffler's solution. And it's an alkaline basic stain, and I know that this actually works quite well to stain the white blood cells, so I'll be using this. And over here I have some some fountain pen ink. In one of my other videos I already showed it to you. You can also buy the ink in those ink cartridges, but it's also possible to have a little bottle here. And for those of you who don't know the ink cartridges, you put them in here, in a fountain pen, and then you can write. No, you won't see it. Here, see? And I'll be using this ink here as well. Okay. As a matter of fact, I can already start preparing this in a second or so. I'll be putting the ink in here. So, how am I going to get the blood? That's I also have a cover glass here. Here. I'll also later on be using this large cover glass because I also want to do a little bit of oil immersion microscopy. But the really interesting question is how am I going to get the blood? And I've got some lancets over here, lancelets, and I got them from a drugstore. And they're sterile, and they look like this, and they're very sharp. Okay, so I have to remove the cover here later on, and I'm gonna prick myself, and then I'm gonna put the blood on the microscope slide. So if if you're not used to seeing blood, let this be a kind of an advisory, a warning that I'll be pricking myself later. Some people can't see blood. Okay, so I'll put it back in here for right now. Of course, some microscope slides. I'll be using over here some of those tips here for the pipette, and of course the immersion oil later on. And over here on the side I have some alcohol, because I'm going to, of course I want to disinfect myself first before I start drawing some of the blood. So I need to open this. Okay. And alcohol is on here. I always like to use my middle finger here of my left hand because I'm right-handed. And why the middle finger? Because I can use the thumb quite easily to press against it, to make sure that a lot of blood is going to come out. Not a lot. A little bit is enough, but I want to make sure that this works fine. So what I'll be doing now is I'll be pricking myself. Okay, and this is now here it is. Okay. And this is now the part which takes a little bit of determination and nerve, because it's mostly psychological. It doesn't hurt a lot, but it's psychological. Okay, so I'll be making two slides, one for each type of stain, and in one of them I'm also gonna be I'll put the cover glass, so for oil immersion microscopy. Okay, so you have to remove the cover here. Okay, and here that's the needle. Okay, very sharp and pointed, and if you think too much, then it's gonna be difficult. So I have to kind of, oh my gosh, okay, this is not easy. Okay, here we go. Okay, here we go. You put the drop of blood on one side of the slide, and because it's going to stop bleeding very soon, I'll just collect the blood right now. Also, you see, it already stops to bleed. Okay, over here, and of course, I want to wipe off. The rest. Okay, here we go. So, what I'll be doing now is the following. I'll be now adding a little bit of Löffler's solution methylene blue to one of them. I'll be using the smaller one over here because the larger one might take a little bit longer to dry out. And and here I've got a little bit of the solution, and I directly put it. No, this was mostly air bubbles. Okay, and that's pretty much it. I like to use a pipette also to mix this a little bit. So you draw it in and out. And I also remove a little bit of the excess because of the methylene blue. There is a little bit too much fluid here. So I'm going to discard this, okay? Because I'm going to now use a second microscope slide to make a very thin film of blood. And that's, now you know why I put it actually on the side of the cover glass, because look what I'm doing. You need the second, not the cover glass, the side of the slide, because you need a second slide. You go over here like this, and you see how the blood spreads apart, okay? And then you simply start pulling it over like this. And that's it, okay? This one I'll discard. And this here now can go directly under the microscope, and we're going to have a look, okay? So I need a place to put the trash. I'm going to switch over now to the microscope view. And let's see if we are a little bit lucky. I've got now the 10 times objective in place. Okay, and I might need to refocus a little bit. And we already see those very slight clusters of red blood cells, still very small. Okay, let's put the alcohol away. Yeah. Okay, here we go. And the dots, these are all red blood cells. And for the white blood cells, I need a significantly higher magnification. So I'm going to up, this is now the 20 times magnifying objective. And also the brightness is not very high. Okay. Yeah, so, and most of them are indeed red blood cells. You see that some of them are still moving around. They're moving because of the liquid flowing. So this is now 40 times magnification. Okay, and now you can see the typical, donut-like shape of the red blood cells. And now we have to find some white blood cells, and I'm now seeing that there are not so many of them, because I remember quite well when I tried this some time ago, the white blood cells really stained nice and blue, and I don't see anything like this right now. I wonder why. I'm going to remove the polarization setting. Okay, so let's have a look here. So you see that, of course, the red blood cells now, because I removed this, I mean, the one in the center right now, but it does not really stain very well. Honestly, I am very, very disappointed, because the white blood cells, maybe you were able to see this here. Okay, I have to go up with the magnification yet further, so I'm going into 60 times. Maybe this one here. This is still very disappointing a little bit, but you see this one cell in the center, which stained a little bit blue, could be a white blood cell, but honestly this is not what I expected. So what I'll be doing is I'll be continuing to look for some of the white blood cells. Maybe I did not use enough stain, but honestly I was really much more successful some time ago. How can it be that I don't see anything? Well, okay. There is another slightly blue spot over there. Maybe I know the reason. I think I know the reason. I simply don't have any white blood cells. This is evidence that I don't have any white blood cells and I'm still able to survive. How is this possible? Okay, I'm just being sarcastic here. Some people might actually believe what I'm saying. Okay. So that's basically one explanation. Look, maybe here. One explanation could be I don't have any white blood cells, or the second explanation could be that there is some flaw in the method. Either I did not give it enough time to absorb the methylene blue, or the concentration wasn't high enough. I mean, you see there are other cells there as well, okay? Or for whatever reason, some other reason. So that's basically a little bit disappointing, but you already know that I will show you a result, because of course I want that my viewers keep watching. Okay, we'll figure it out. Well, okay. Well, before this sample dries out, I'm going to repeat the whole experiment again, and I'm going to increase the amount of methylene blue and also the time. So to give it a little bit more time to absorb the stain, and then I'll simply draw another drop of blood later to test the ink here. Okay, so okay, 1, 2, so that's a little bit more, and I will be mixing this here again. So maybe it was not properly mixed before, I don't know. I need some kind of a new tip. Okay, and let's give it a try now. Okay. It could also be quite well possible that the place that I observed on the slide, was a place where the stain and the blood did not mix well. Okay, because it's so I'm going to increase the mixing significantly more. See, way too many bubbles, So I'll be putting this here away. Look, this is how I discard it. That's it. So this also removes the bubbles here. Yeah, sometimes those experiments can be quite educational in the sense, also for me, because I also learned quite a bit here. Okay, not everything is successful in the first try. And when I remember when I was still at working in the lab at university on my master's thesis, well, most of the experiments I had to repeat because they didn't work. Something had to be optimized. So so this was actually now much more mixed. I now mixed it much more, and I also used more of the stain, of the methylene blue stain. So let's try it again. Okay. Here again, let's go like this to collect everything properly, and then let's go. Okay, so let's give it another try. I switch over to the microscope view. Let's remove the old slide here. And I'm not going to change the focus now. I'm going to leave it at 60 times because everything else should be the same. And let's go directly in here. And a little bit of refocusing might be necessary. Okay, and let's give it another try here. You can see that, the red blood cells don't look donut shape anymore. I guess they absorb, because of osmosis, they absorbed some of the liquids, and the density is significantly lower. Ah, yes. Okay, look at this. Okay, so here we go. We are successful. Okay, so this here is a white blood cell, and I need to explain it a little bit. First of all, I know it's white blood cell because it stains blue, and you can see the nucleus. That's the dark shape on the inside. It is a little bit kidney shaped. So it's basically many nuclei not many nuclei, but the nuclei of some white blood cells are formed in a very irregular way. That's why they're called polymorph nucleated. Polymorph means in many different shapes, and depending on the shape of the nucleus, you are able to kind of categorize the white blood cell. Yeah. So over here you see two lobes. Yeah, and I already forgot. I don't know. There are the eosinophils. They are there. The basophils are there, and so on. And depending on the shape of the nucleus, you can categorize the white blood cell. Okay, you see that they're a little bit bigger than the red blood cells here. Okay, yeah, and I keep on looking. And you know what? I'm going to go down with the magnification now because I simply want to get a wider field of view. Okay, and here, this was basically one of them. Here, look, is another one. Okay, you get the point. So up to 60 times again. Okay, here we are. Need to increase the intensity here. Polymorph nucleated neutrophils. I love this word. Okay. They have, I think, 3 to 5 different lobes. Yeah. And then there are some cells that are basically have a completely round nucleus. So, I mean, there are many different, kinds of white blood cells. And of course, for those of you who are interested a little bit in statistics, you can now make a white blood cell count and how high is the density? Here we go. How high is the density of the white blood cells in relation to your red blood cells, and so on and so on. Yeah. Yeah, I think here you again see quite nicely that the nucleus, the dark blue stained part in the center. In this case, I see over here 3 of them. And if you look very carefully, then you're able to see that they're actually not completely separate, but they're connected. Okay? they're connected together. Three lobes. Okay, I don't know what that is. Yeah, here's another one. Okay. Yeah, there are two lobes. And so on and so on. So you basically can do a little bit of chasing white blood cells here. Another one. Oh, okay. So basically take-home message is the following. Yeah, you got to simply give it enough stain, and you also have to give it a little bit of time mixing, and maybe it's a good idea to really mix the stain and the red blood cells and the blood together, because they do have a different density after all, and so it could be that diffusion alone might not be enough for the stain to reach all of the parts of the blood. And yeah, here's another one. Okay, you get the idea. Okay, so what I'm going to do now is the following. I'm going to repeat the whole thing, and I want to try. That's what I say. You got to be careful that you don't drop the bottles. Luckily, it's closed. Yeah, I'm going to repeat the whole thing again, and I'm going to now use regular ink. Okay, and let's see how lucky we are. And then later on, I might go back again, and I would like to look at the methylene blue stain again, but using oil immersion. Okay, so that's kind of my the thing that I'd like to do. I'm running out of slides. I'm just realizing. Just going to reuse those again. So, oh my gosh, what does this mean for me? This means that I have to draw again a little bit of blood. Okay, so let's try it again. This is again a little bit of psychology that's necessary. 3, 2, 1. Ah, this was deep. Ah, okay. Here we go. Okay, here is one, and you know what, I need another slide. I'm going to use the possibility right now to simply put some blood also on another slide. Very quickly I have to wipe it, because out of the box they're rarely clean. Okay, again it's a microfiber cloth, and here we go again. Hope there's still, yep, ooh, okay, fine. So, what did I say? Yes, I wanted to use a little bit of ink. So I'll be using this here as my little ink. No, you know what? I'll do it even simpler. Look what I'm doing here. And here we go. Goes back into the fountain pen. Yeah, so I didn't even need this here. And of course you want to exchange the tip again. And let's do again a little bit of mixing. So, again, it's quite foamy. I think that should do it. Okay. Okay, that's the waste. By pressing this button over here, you can throw off the tip. Look. Okay. And no, this was a bad idea. I just realized I did this too early. I wanted to remove a little bit more of this. Okay, a little bit more of this, because it's still too much. So I'll now be using the other side of this one here. It's sticking together because the blood already is starting to coagulate. So I use the other side here. And same story. It stains much darker. I don't know if this is not going to be too dark. Okay, I have no idea. But yeah, that's the whole point of science, is also to do a little bit of experimentation. So I'm going to switch over again to the microscope view. So let's remove this here. I've got the 60 times magnification still in place. And if this doesn't work, I think it might be indeed a little bit too dark. And too liquid. Too liquid as well. Okay, too liquid as well. But something is staining here. Actually, it looks kind of nice, I have to admit. Yeah, I mean, there are certain things that kind of stain blue in here. Do you see this? But these cannot be white blood cells, of course. They don't look like it, and there are way too many of them. Okay? Way too many of them. So, this is a little bit strange. Yeah, there seem to be some other impurities in there that stain blue. overall, I think the shape of the white blood of the red blood cells looks kind of nice. Yeah, because they are able to move around more freely, and apparently, and that's another interesting observation, apparently the ink does not seem to osmotically change the red blood cells so much, because the ones that you saw before, they were kind of they kind of lost their donut shape, and so it's a question of osmosis. And here they seem, with the ink, they seem to retain the shape better, and there are lots of things that are also stained, but I don't know what this is. Okay. Must have been some kind of a residue left on the slide that actually stains blue. So I think I'm just going to stay maybe with the methylene blue. Yeah, see? Considering the fact how many white blood cells I could see in the with the methylene blue solution, very few here. Maybe I'm overlooking a few of them, I don't know. Yeah, but I mean, I think you get the, get the idea that not all stains are always very suitable. I mean, first of all, the Löffler solution is highly alkaline, and some of the white blood cells are called basophils, and they're called basophils because they like alkaline stains. So maybe the ink that I'm using here is simply not, it doesn't have the right pH, and therefore maybe it doesn't stain as well. And all of those other blue spots that you see, again, I think this must be some kind of a residue on the slide that I did not properly clean away. Or maybe could there be something over there? I don't know. Yeah, so, okay. So either I'm going to try to optimize this a little bit, but I think maybe I overlooked something. I think I was not very successful with the fountain pen ink. Please do try it out at home and post something in the comments, okay? What I'll do now is I'm going to now try oil immersion as I promised, and I'm going to now use again Löffler's solution, the methylene blue solution, okay? So let's switch back to the desk view over here. So let's clean up a little bit. I need this here. I don't need this here, and I'll now be using a cover glass. So where is my methylene blue solution here? Let's give it a nice drop. Okay, maybe another one. Again, a little bit of mixing. I mean, the blood was sitting here on the slide now for a few minutes, so it already started to dry up a little bit, and this might also impair the mixing ability here. So, and when using a cover glass, really important. You really want to make sure that the film is very thin. So this means we have to use very little liquid. Okay. Okay, so very little liquid to make sure that the film really spreads very thinly, because I also want the cover glass to be very flat on the sample, because the working distance between the microscope slide, the cover glass, and the objective is very small. And if the cover glass is now floating around, then I might actually impact it's not going to impact really, but the oil is going to push down on the slide, on the cover glass, and then the liquid starts moving around. It's very unstable. Okay, so I'll leave it at that. I can take this again away. Just remove some of the bubbles as well. Okay, and I'm gonna give it a try now. So where is my cover glass? This one I already did clean. You might have a problem seeing it a little bit. Okay, so I'm going to put it on here now, and here we go. And you see that it spreads nicely. Okay, and let's have a look. Okay, so let's remove again the old, not the old one, but the previous one. Still nothing here. Let me give a last look. I don't know. It might also be that it's kind of difficult to see because of the other stained contaminations. Okay, so now I have to move back to a lower magnification a little bit, because I need to refocus everything because there's now a cover glass on top of it. Okay, here we go. Yeah, see, it's still flowing. I need to focus. And you see that the red blood cells are completely round again. But you see, on top there is a white blood cell. They're completely round again because of osmotic issues. So look at this here. This might be a white blood cell here. Okay. So let's go up to 60. Yeah, they are difficult to see if there is a few of them clustered together. So maybe we'll find a place where there are... look, there is also something going on here. Okay. So, I want to find a stationary one, but this might actually also show you a little bit one of the disadvantages of using a cover glass, because the cells, they don't dry up as easily. Here we go again, okay? The cells don't dry up as easily, and therefore it stays liquid, and therefore there is more movement here. Okay, I think there might be two of them next to each other. But you see that there are also many other things that kind of stain blue in this sample here. So this is now 60 times. Also over here there is one. And here is one that has, again, multiple nuclei. Now it's standing. Okay, and this large cluster, blue cluster that's next to it, I don't know what that is. Okay. Yeah, here again, it's moving along. Yeah, but I'm looking for one. Here is one. I'm looking for one that actually has multiple nuclei. There is one here in the cluster of other cells. Okay. So what I'm going to do now is I'm now going to go up one magnification higher, a step higher. So, and I'm going to now do the following. I'm going to now use oil immersion. I'm just going to show you here. This is immersion oil. It's synthetic immersion oil. I'm not a fan of this because it requires me to then clean again the microscope objectives and so on, and it's messy, and you have to be careful that the oil does not touch the other objectives. So, but still, for the sake of this video, I'm going to be using this, and I'll be just looking at the cell that I see, you see pretty much in the center now. Okay, so I need to move over a little bit. So I'm going to rotate the 60 times objective out of position, and I'm going to now put a drop of immersion oil on it. No, you know what I'm going to do? I'm going to do this differently. I'm going to actually apply it here on my table, and then I'm going to put it in. Okay, so I'm going to take it out. Okay, here it is. So, and here, right on here, I'm going to put some immersion oil. And now, don't use the 60 times, very important. I have to switch over to the 100 times objective. Just a second. It's a risky thing. And I hope it makes contact. Move it back and forth a little bit to spread the oil. Yeah, and as I said, the immersion oil pressed a little bit down on the cover glass, and therefore the things start moving again. And but no, it's still in place here. Very nice. Okay, here we go. By the way, this is all what I'm doing right now. This is all in bright field. So here we go. Okay, so this is now using 100 times magnification objective, and we now nicely see this white blood cell over here, still stationary, kind of captured in between a whole bunch of other cells or debris or whatever. I don't think these are other cells, cannot be, because if the cell clusters in the blood, that would be thrombosis. You don't want that. So I suppose it must be some kind of an impurity here. Yeah. Okay, there might be also over here another cell. Yeah, again, so we're now basically at the maximum magnification that we have. Yeah. And maybe we're lucky. Yeah, here's another one. No, actually, it's easier to see them, so maybe it does take a little bit of time for the cells to actually absorb the stain. Yeah. Yeah. Did we just have this one over here? It's not very clear, this one. Okay. So, and there's one over here as well. You get the idea. Okay, here we can see the multiple nuclei also quite well. On the top left corner there is one right above my head. Okay, here is another one. There are two lobes visible here. Yep, there are, yeah, here on the right side. Here, there are two, yeah, the one on the bottom was already the one from before. There's one over here. Yeah, above my head again, one with a round nucleus. Okay. There seem to be two of them here, difficult to see a little bit. You get the idea. Here, another one. Well, there are lots of them right now. Here there are two of them, on the top and on the bottom. Again, a whole bunch of cell clusters. Yeah, there's one over here with this, yeah, it looks a little bit kidney-shaped, the nucleus of the one. There's a very dark one over there. Again, a strange cluster. Three of them. Okay, I'm quite, I think you get the point, okay? Take home lesson, don't give up. If you do not see something right away, yeah, think through what could be the problem. Give it a little bit more staining time, maybe, okay? Yeah, and I'm not saying that the fountain pen ink does not work at all. It could be that simply maybe I also didn't give it enough staining time, or maybe there was some other problems, or maybe it really doesn't stain, or maybe it's really not suitable. This could quite well be. Okay, don't give up. Always change one thing at a time. Like I did right now, I simply mixed it better. Yeah, and it could also be that in the first try where I was not successful, I simply looked at the part of the slide where the methylene blue did not reach it properly. This can also be. Yeah, here's another one. Okay, a whole bunch of clusters again where I don't know what they are. Yeah. Okay, enough for today, I would say. Yeah, I hope that it was again a little bit informative. I'm just realizing that here on the other side here, there's a little bit of a, I have to adjust my background a little bit. I hope that those little imperfections in my videos do not matter too much. Okay. Yeah. I hope that in any case that this video again was a little bit educational, as just a last warning, not warning, but simply a comment, because there might be some teachers also watching this year. I myself am a teacher. Be careful. You're not always allowed to take blood samples in a classroom. Depends a little bit on where you live and what the legislations and the laws are. But always when taking blood, you have to be very careful, and it might not always be allowed, because this way you can of course also pass on infections and other problems. Okay, and yeah, just a short disclaimer. Happy microbe hunting as always, and see you around next time. Bye bye. Okay, yeah, cleanup time. Yeah, this stuff I'm not going to be reusing. I'll just throw it away. Sometimes I do reuse old slides. I just rinse them, but actually I'm not going to do this now, especially with blood samples. Well, okay, here I'm just also gonna, before I throw it away, I'm gonna put the thing here again on top. Yep, and I need some tissue paper to, not tissue paper, lens paper to clean my microscope. Okay, so all of this goes now, this here as well. Yep, all of this now goes into the trash can.